The last homework was use your language. Maybe you use PowerPoint. There are some video typing functions in there you can record. See, what is your understanding of matrix multiplication? How are you going to do it if you have two matrices? And what that means, basically. Why it's important? Let me bring you back to news. So this is a news brief. Let's see this. It's about sometime last year, and Google, using their AI, discovered a new way to do multiplication of two matrices. So this new way will save some of the intermediate step not involved in the multiplication operation, but replace one or two multiplication steps with addition. So this will save a lot if you do many matrix multiplications. Why this kind of news can come up to the headline of those news stories everywhere? I don't know who pay attention to that. Any one of you know this? Nothing. So basically, if you do multiplication, the standard algorithm, you know how to do it manually. There are shortcuts, and there are this human established algorithm will save some of the steps, but the Google machine learning algorithm discovered a new way to do it. Now you see all this and the Nature News. So this is a big story because this matrix multiplication involved in all the machine learning, data science operations. If you streamline a video and it's only frame of the video, it doesn't involve matrix multiplication, but a lot of other operations you need that. We will have this. So this week, is already week 3. We are going to discuss distance, okay, different kind of distance you can use to measure the similarity or dissimilarity between data points. We will go back to the inner product, okay? You have done this. You have two vectors. Once you take inner product, you get a scalar, a single number. And we will revisit this operation and see what that means and geometrically. Then follow that, we will define cosine distance. You probably know Euclidean distance, but cosine distance is also widely used in machine learning, especially in high dimensionality analysis of data. You don't have to compute Euclidean distance, and cosine distance sometimes give you a quick way to compute the distance between data. You will see cosine distance much easy to compute. It's dot product. So that's why it's widely used. That's one of the reasons. Then we will look at Euclidean distance matrix. And I will explain that based on this matrix, you can do so-called multi-dimensional scaling. Okay, so you use the pairwise distance between all your data points against all data points, and then you can reconstruct their distribution in high dimensionality space, but then you can reduce the dimension to look at their relationship. At the end, we may spend another 20, 30 minutes to revisit cell ranger report and go a little bit of detail and see how should you set cutoffs and get rid of genes, cells, low quality, what's the cutoff for mitochondrial ratio. Okay, any questions before we start? No? I will use 3 slides to recap if you still remember what you learned from high school, geometry, trigonometry. So this is a cosine definition. And the cosine, and if you have a triangle, then you have a theta is one of these angles. Then this is a perpendicular, and then this is called adjacent, and the ratio between this adjacent with hypotenuse is cosine. Okay, cosine definition. You need to remember this. Another definition is this Pythagoras theorem. This is even easier. Everybody should remember. The beauty of this theorem is you can apply this to higher dimension. In this triangle, this is certainly 2D, right? And this particular triangle, if you have 4, 3, and you know this diagonal, this perpendicular is hypotenuse is 5, right? Once you calculate 5, and then the height is 2, you can redo this again, square root 5 and square root 2 and square root of that. You give the length of A to D, and then you can put everything together. You can have 3, 4, 2, and put 3, 2, 4, and square root, and get the 2 squared plus 3 squared plus 4 squared, in order to calculate the distance between AD. Draw that again. So you have this 3D let's see, 4 here, 3 here, and 2 here. So this is A, this is D. You just apply Pythagoras theorem twice, then you get the distance between AD. As I said, this applicable to even higher dimensions. So this is only 3D, but if you have a 4D, 5D, you just keep adding your terms. You get the distance between 2 data points in higher dimension. Okay? No question, right? Here, it's easy. Another, this is a rule based on the definition of sine and the cosine. We are going to use this a little bit late here to derive the cosine distance formula. So I just need you to remember, for example, one of this. Okay, if you have cosine, okay, one of this mu rs. So if r is minus s, if you have 2 angles and you found 2 angles, one minus another, then the relationship between these are cosine r cosine times cosine s minus plus s I think rs RS, RS. So we are going to use this relationship later to plug in a formula, but this can be derived using the definition of those, okay? We also introduced unit circle last time. So this gives you more example data points on the unit circle. This 0, 1, 1, 0, certainly. 0, 1 is on the unit circle. The definition of the point on the unit circle is its norm is 1. The norm is the length, how long the magnitude of this data point. If you have a length is equal to 1, then you are on this circle. So if you use plug those numbers in to calculate the norm for those vectors or this data point, they have all the norm are equal to 1. If you have a vector, see it's not on this unit circle, see this new, okay? It's outside here somewhere. What you can do is this vector contains two numbers, right, in this 2D. It's x and y coordinate value. What you can do is to divide this vector by its norm, divide by how long it is, these two numbers, each divided by the magnitude. That will bring back this particular data point back to the unit circle. So that's an operation. That's an operation you want to know. We will do a lot of this. So that's a mathematical formula. In MATLAB, your data point vector divided by its norm. Then that vector come back to either this unit circle or unit sphere or even high dimensional hypersphere. Same operation. Why we want to normalize this? So people use normalized, people in biology use, in mathematics people use, in data science use normalize. So that's what we mean by normalize a vector. You normalize, you bring it back to the unit circle. You don't keep this scaling factor. You normalize by a scaling factor and bring it back to the unit circle. Only keep its direction. Okay, so that's the purpose. You normalize your vector by its norm, bring the data point back to unit circle, get rid of the scaling factor, keep its direction. Okay, so you know how to multiplicate two matrices, A with B, term which Z. Okay, the term between x, y, between xz and zy has been zeros, so they are not show up over there. So this is one way to understand the dot product piece by piece. This should have a start on this slide, okay? We have a star on this slide because this is one of the most important formula we will use. This formula tells you the dot product of two vectors, two data points, okay? In this 2D example, mu and nu and their product equal to the norm of the first times the norm of the second and times cosine of the theta between these two vectors. We know if you have a vector, the dot product is a single number, okay? So this single number, its value the same as the length of this times length of that times cosine theta. So this is a single number, this is a single number, this is a single number. So the product of these three is a number, okay? So your left is a number, your right is a number. So I'm going to approve this is true if I'm going to use example to show you, okay, this is true based on the definition. You know the definition of this. This is how long it is. This is how long it is. And we know the definition of theta. Then we see whether this is true using my small example. Just convince yourself this is okay. So I keep this because we are going to use different ways to derive this formula using this relationship in different ways. Okay, so that's the formula, the example we want to use. Now I put a special mu. This is special mu in order to simplify my derive of formula because it's in the x-axis itself. So this particular vector mu, I gave its coordinate. Okay, you know if it's vector, is writing in this way x mu and 0. I save a variable because I put it in x-axis, so I can put a 0 here. Otherwise, I got a little bit messy, but in this case, I want to do this. And we also know this vector, how long it is. So that's the length. How long this? This is the norm of it, okay? And this number also numerically equal to x mu. So far so good, I think. Everybody agree because I rotate my vector and let it to be on the x-axis, so I can simplify my notation like this. Well, the mu, another vector is more generalized, so it's just like this. Mu. It's also defined by two numbers, x mu, y mu. Okay. So by definition, the length of this is it's a norm. Okay, now we need to know the definition of the theta. So what's cosine theta? So cosine theta is, if you do a projection, basically you project your new vector onto mu vector, and then this is 90 degree, and by definition, your theta should be, the cosine theta should be this. Oh, by the way, the length of this blue is equal to xv because that's the coordinate of the nu. So we know how long this part is. This is the value is x. It's same as this, right? Because that's where the position, the coordinate position of this is. And this divided by that is cosine. So x divided by mu. That's by definition is a cosine theta's definition. So that's why we can write this equal to its norm cosine theta. So this x, so this value here, x nu equal to cosine theta times its norm. Is that right? Okay. Now we can put them together. So let's see. Mu this two dot product is, let me put that 1, 2, so put a dot product, so x mu y, and put that one here, x mu 0. So that's what we want to compute, this dot product is this times that x v x plus y times 0. So that's the way you do the dot product between these two vectors. So numerically, the number of this product is, this is 0, okay? It's actually x mu, x mu. Is that what we write? Yeah, so if you do that, you get these two numbers product. And then you replace x mu. You replace x mu using its norm. So this becomes and then you replace this with that. So it becomes the cosine. Okay, I just do some basic formula here and replace some numbers, follow the definition of each of this. Okay, I just use this special example to convince you that's true. This product, dot product of these two vectors is the norm of the first vector times norm of second vector times cosine theta between the angle of these two vectors. Okay, this. So that's another way to do this using the formula I'm writing here. So instead of using the rotation, it defines two angles, r minus s. So if you have these two directions, r minus s is angle between these two, and then you apply this formula, you will get the same answer, which is this. Okay? And there another geometric proof of dot product, and also reach the same conclusion. here, A and B are two vectors, and AX times BX becomes the area of these two square. Then somehow this proof is true. Okay. Two rectangulars. Okay, so you remember this formula. Then you need to know what's the application for this. The first application is to calculate the angle between two vectors. Because you know mu times v is mu norm times v norm cosine theta. So you rearrange this, then cosine theta, the angle between these two is that divided by mu nu. Okay, so if you don't know this angle between these two vectors, you just do a dot product, okay? And this become a single number. And then you divide by the norm of the first one times norm of second one. Then you get the cosine theta value between these two vectors. Why is it useful? If you reach conclusion that the cosine theta between these two vectors is 0, means that they are orthogonal to each other. Because when theta is 90 degrees, then cosine theta is 0. So if you get a 0, you know the dot product is 0 or whatever. You know these two vectors are orthogonal to each other. Another application is you can compute the projection. Projection is this like this. So if you use B as your the vector you want to project A onto it, so you shed a light on from this direction. Okay, you project your A onto the B and create a shadow. So we want to know how long this shadow is. It's from this position to here. By definition, the length of this is the norm of A times cosine theta. Okay? So if you rearrange this formula, you see, okay, length of A times cosine theta equal to the dot product of the two vectors divided by the norm of the B. So this side is easy to compute. It's easy to compute A and B dot product, easy to compute the norm of the B. So the ratio between them is how long this projection is. So this is a very quick way to compute the projection, the length of the projection from one vector to another. Question, yes?
I was just curious about, so when we have the data, how are we going to apply this, like data analysis of, let's say we have a cluster.
Yeah.
So we will have to, I mean,
I can show you one example of this projection. For example, you know the coordinates of A, so because it defines x and y axis the coordinates. But if you say, okay, I want to use B as my new x-axis. So you want to compute the projection of A project onto B using B as your new axis, x-axis. So what's the new definition or new coordinates of x and y for the x onto B? So that's one of the applications. So you select, rather than the original x-axis, which is horizontal, but you want to use this, the B with the angle as a new axis. You want to project your data point onto the B in order to see if I use the B as my new x-axis, what's the value of the A's coordinates using B as your new x-axis. So this is like one of the applications. So you can compute this very quickly. Just use products and the division, right? And another is this formula is used in compute the distance between a data point to a line. So if you use B as a line, and okay, and you can compute the distance from here to here, which basically you want to compute this part using this formula very easily. The application, as I described, the changed coordinates. You use your new, this selected coordinate system, use B as your new vector, new x-axis, and you can compute what's the new values, coordinate values of A in terms of B rather than the original. Is that clear? Maybe I use one example to show. Is that clear now? Somehow? Again, maybe one small example to show that. So that's my current coordinate system. So I have x-axis and y-axis. I have this beautiful A vector here. Okay, so what's the coordinates for the A? So you have a value which you project this onto x-axis, y-axis. So this will be x A, y A. So A has this old coordinates x A, y A. Now I'm going to use B. Basically, I want to rotate my x-axis to some angle. I use B, this as my new x-axis. This is my B. I'm going to use this as my new x, and my new y should be somehow here. Now, if you use this as your new x-axis, the coordinate of A will change. It's not xa, ya anymore. You have to compute two values regarding this new axis. Two values for A should be how long this part is, right? and how long this part is. That's a new coordinate for A position. But how can you compute its new coordinate here? That's a projection of A onto the B. So we need a quick way to compute it. So what's the quick way? The quick way is to dot product A and B, is that right? Divide by the norm of B. Okay, so you quickly get this coordinate, new coordinate system. You want to compute this very quickly because you want to try a lot of this new selected direction of the as a new axis, as a new x-axis. Okay, so we can use cosine theta as a similarity calculation for two vectors. If they are orthogonal to each other, and then 0, right, the distance. If they are somehow towards the same direction, this value will be higher. Okay, if they are same direction, then you have a similarity equal to 1. So this angle gave you a quick and dirty way to compute, especially the data points in the high dimension. Scaling is really not important. It's really how long, it's direction, the angle between them is important. So you use that as a similarity. If these two data points point to the same direction, then they are the same. So 1 minus cosine similarity is a distance. And if you look at it carefully, and their relationship between cosine distance and Euclidean distance, if these two data points are on the unit circle, then their Euclidean distance is the same as 2 times of the cosine distance. So this is some kind of formula between that. So you see a lot of the terms, like this y vector x divided by the norm, basically is to drag it back to the unit circle. And once you do that, the Euclidean distance is the same as 2 times of the cosine distance. So we are going to use this projection technology in PCA. In PCA, remember you have many points, like A, right? Now, in this case, we only have one small a. This is one sample. But in reality, you have a lot of a. So you want to rotate this x-axis many degrees. Theta could be 0, 1, 2, around this. You want to find a particular angle of a B to let all your data point A project onto it. Then you compute how dispersed they are on the new axis. Okay? If they're separatable, become more separatable because their coordinates become longer from each other, then you select this as a new axis. Something like this, if you have an original data set like that, you need to have a new axis angle. So allow this data point to project onto it, and then they are more separatable. If you select the wrong angle and then project out, all the data points cluster and together, it's not. So in PCA, we will need this technique to do the projection. So that's why you need to do this quite a lot. As I said, we can do data point to a line here. So this in the SVM, then you found a line which cross between blue and the red data points. So try to separate them as much as possible. So in this case, you want to compute a lot of this distance from data point to a line, because we want to select different lines. You have angles, you have intercept in order to move the line to achieve maximize the separation between these two clusters of data. So you need to compute the distance. And this formula, okay, it's from one data point to a line, and use this relationship as well. So we will discuss this later and in SVM, and then we are not going to do this now. Okay, some quick definitions of orthogonal. So if you have these two vectors, their dot product equals 0. Okay, so the dot product oh, go. So for the vectors, these two shouldn't be 0. You should have some length. And then in order to make a dot product become 0, you have to have a 90 degree between them, theta, cosine theta is 0. So that's why if you have a dot product between two vectors, get the result 0, then you know these two are orthogonal to each other. So why you need to have these two orthogonal vectors? Because you can use them as a new coordinate system, as a pair of x and y axis. The original x and y axis is like this, 1, 0, 0, 1. But you can rotate using this set, this blue one, as a new coordinate system, as a new x-axis and a new y-axis. So you can decide or compute orthogonal vectors, a pair of this, and use as a new coordinate system. This also applies to 3D, and if you have combinations of this, okay, point to some direction in 3D, and this coordinate another direction, and you compute dot product between them, they're 0, and you know, okay, this is a set of this x, y, okay? And then you can add another, found another. They also, this one, the third one, also have dot product equals 0 to this and to that, so you know 3 of them are 90 degree somehow. So you use this as a new coordinate system. So this set is called a set of the orthogonal vector set, and then you use those orthogonal vector set or orthogonal matrix as your new coordinate system. If it's 2D, then you have A, B, C, D, 4 numbers. If it's 3D, you have 9 numbers, and each column point to one direction, like this, okay? If it's 4D, you have another dimension. if those vectors not only orthogonal to each other, it's also normalized. Normalized means the position of this vector divided by its norm, so it's back to the unit circle or sphere. So this vector is called orthonormal vector set. So this is like a standard coordinate system you can use to define your new coordinate system, orthonormal. If you have orthonormal, orthogonal matrix, the multiplication of this matrix by itself, okay, you get an identity matrix. This is like an example. Okay, this system is also called unitary. Like 3D, you have a unitary rotation matrix. Okay, we use a lot in quantum computing because in quantum computing you deal with this sphere and you have a electron spin and towards a different direction, either up or down. This is 0, this is 1, and in the middle is superposition, and then there's the phase. So you use a lot of those unitary rotation system to describe a state of the electron or super conducting qubit or something like that. Okay, linear independent. So basically, if you have a system which you cannot use the combinations of other data points to reach the third one, then you see, okay, these three should be independent to each other. Otherwise, linear independent, dependent means you can somehow use the combinations of these two and reach to that. So basically, you add 2 times of this, 1 time that, you reach that, right? So this looks like a 3 by 3 matrix, and same here, 3 by 3 matrix. Internally, and this is a linear independent components, make this matrix informationally more rich or contains more information compared to this, because this you can reduce to some combinations of the components and then reach the third one. Okay? So this introduces another concept called the rank of the matrix. So in this case, this matrix has rank 2 because you only need this first and second column, these 2 vectors. Their linear combination can become third, so you don't need a third. This is like some kind of redundant. However, you cannot do this for the second matrix. So the second matrix have a rank 3. So rank give you a value to look at the internal independence of the data points. Or you have a higher rank means all the data points are not somehow related. They're on its own and occupied some space by their own direction. Otherwise, you can use several combinations linearly to reach the others. So let's see. In physics, people use that a lot. Yeah, question?
How do you come to know whether it's redundant or not? Like how is the ranking system based?
How the rank is doing.
How they compute, like how they give rank?
Yeah, how they rank. Yeah, I don't know. It is numerically how this function work and to reduce the dimension, how they search for this space. I haven't read. I should know because they're numerical linear algebra. Those books will give you the algorithm. See how can you compute a rank of matrix.
How did you say that it has rank 2 and because the numbers are same, this placement is different. So can you explain?
Yeah, I will show you later.
It's the number of linearly independent columns.
Yeah, I show you later. Yeah. By the way, the rank has been always used quite a lot in physics or data science to indicate the level of complexity in your data. So this here, low rank hypothesis basically they say, okay, some system network looks very complex, but in reality, you probably can just use a few components to reconstruct everything. So what that said is something phenomenal looks complex, but in reality, the components, number of components are limited. So you can reconstruct this very complicated world with this limited number of the components. So this is called low rank and hypothesis. Come back to this example, and what you can have is this column is somehow redundant because you can use combinations of two of the column 1 plus 1 column 2 and become this. Okay, where you cannot do this for this matrix.
So low rank means high redundancy.
Yeah.
High rank means low redundancy.
High rank means you need more dimensionality to describe your system.
High redundancy doesn't mean you can delete it if it's not zero. If your data set is, say, all 100.
Yeah, it's not deletable, but the complexity of your system can be estimated by the number of the rank. I think this is another example. Let's see. This example gave you some geometric insights of what the rank is. So this matrix have two values, two vectors. But these two vectors only need one single line to describe it. You don't have to expand this to higher dimension because the rank so what's the rank of this? So you cannot do, this should be 2, right? Yeah?
Should be 4, 4. Because there are no non-zero rows of columns.
Oh, then the rank is like counted by the smallest either column or.
So it's mentioned that like they calculate depending on non-zero
Yeah,
columns.
they said rank of the matrix is calculated by counting either non-zero rows or non-zero columns.
I'm not so sure. Non-zero. If the non-zero means the whole column or whole row is not zero. If the whole column is 0, we should remove it first, maybe.
Yeah. So
Okay.
in the previous
Mhm.
rank of matrix slide,
Mm-hmm.
it has rank 2 because one of the rows is completely 0.
Mm-hmm.
So the rank is 2, and then when there is no 0, the rank is 3.
Okay.
So I mean, I didn't know this, but that's what I said.
Okay. You mean here because it's 0.
It is 0, so it says we either
Mm-hmm.
calculate all non-zero rows or
Mm-hmm.
non-zero columns, and that will be the rank.
I'm not so sure. Maybe in this case, both column or rows are both are rank 2. I don't know. Let's double check. Okay. Mm-hmm. Thank you for Google for that. Maybe it's because this is no information, so you need
Yeah, they
rank
say,
2.
any non-zero row will be counted out.
Mm-hmm.
That will be there.
Okay.
Sorry,
You can delete. Yeah, you can
any
delete
row
it.
containing all zeros will be counted out,
Mm-hmm.
and that will be the rank. So that means if it has low rank, that means higher redundancy, right? Because no information.
No information there. Yeah.
After you delete, it becomes higher rank. If after you delete, it then becomes rank 3 because you remove the redundant.
I think the rank is based on either row or column, and which one is smaller.
Yeah.
Smaller, yeah.
So
It's still 2. If you delete this, it should be 2. Be 2, yeah. Okay, so good. So we can play with some examples maybe in the computers and see. Okay. Well, it's time for break. Say 5 minutes, okay? Yeah. We come back, I don't have a number. 6 past 11. Okay, let's continue. Because this also normal base matrix is so important because it can define a new coordinate system. So their function in MATLAB and another language also, and you can see, provide me a set of vector, and if you 3D, you gave 3 vectors, say, okay, this, that, and the different lengths. And this function will take that vector site and try to keep one stable and keep another, and rotate it, make it orthogonal to the first one, then make the third orthogonal to the first 3, and return you a new orthonormal matrix to allow you to use this as a new coordinate system. And if you gave, say, like 3 in the same plane, then it probably will just give you x, y, because they're no necessary to have another dimension because everything is in 2D. So it's based on the rank of the matrix. This procedure is called the Graham-Schmidt process. If you look at the source code, it's pretty easy, and some normalization, the dot product between elements. But just give you an idea, okay? You can quickly get this if you have data and you want to construct a new coordinate system based on your data points. Yeah, not the best, but based on the data, the 3 data you gave, it will construct a coordinate system which 3 vectors are orthogonal to each other, allow you to use them as a yeah. The one you gave to it, not orthogonal to each other. The algorithm will you drag and use a force to allow it to become orthogonal. Okay, the Euclidean distance between vectors. This is easy to compute. You just take a norm, and you use data point 1 minus 2 and norm it. Okay, that's the distance between 2 points. This is an example. So if you have x, the coordinates for this, the value is x1, x2, y is y1, y2. Distance between them is, okay, y1 minus x1 squared. But this one, and at the end, is just x minus y or y minus x, then take a norm. The way to write in this is you don't have to go into the vectors to compute individual values. You just everything together, okay? Especially if you have a vector in a high dimensionality space, then this vector is really long. It could be hundreds, thousands of vector points instead of just x1, x2, maybe very long. But you'll still be able to compute the distance between x and y, just minus together and take a norm, right? You don't care inside what the value are, as long as they're same length. And you can do this if you have a vector set. So you have 5 points here and the 3 points here. So you can compute a matrix from this each of these 5 points to each of these 3 points. What's the distance between them? So that's a pairwise distance matrix. let's see, A, B, C, D, E to A, B, C, small a. So you compute a matrix, and then this is the distance between big A to small a. This distance between big A to small b. So you fill it up. And you know how to compute the distance between those. So that's just, so this D, for example, D, big B, small b, is a norm of big B minus small b. So you can compute this very easily, okay? In reality, you don't even have to compute each element by element. You can do this matrix operation to get those. The matrix operation explained somewhere here. We will use a computer session to play with some of those, okay? Because each of this is a vector, okay? A is a vector xa, ya. The big B is, so this forms a matrix, this forms another matrix. So you want to compute the pairwise distance between data points. Their matrix operations can help you very quickly get this. But you know you can do this element by element. You can use this minus that, take norm, you know, you fill up this. It also works just a little bit slower than the matrix operation. So why we want to use this distance matrix? Especially, the very useful one is not consider another cloud of data point, but itself. So you have A, B, C, D, E, 5 data points in your space. Then you want to compute A, B, C, D, E, the squared matrix. So this is pairwise distance matrix between data points themselves. And the distance between themselves is always 0. So you have in diagonal position always 0. And this just like a map. This is like from one city to another. What's the distance between them? Then you see this in a train station somewhere. Our problem right now is if you are giving this Euclidean distance matrix, can you reconstruct the map? So if I gave you this matrix, tells you from Lausanne to Geneva is 33 kilometers, and from Geneva to Zurich, 158 kilometers. Would you be able to use this to reconstruct the map like that? Is that possible? Do we have the intention or interest to do that? I think we do, because if you do this cell distance matrix, you compute one cell and the distance with another, you get this huge matrix. You want to know whether I can build a map between cells. So I know from this cell to which other cells is close to my neighbors. So at the end, we can make a clustering algorithm to put the cells in the cluster together by their distance using this map. Turns out you can somehow, using multidimensional scaling to build a map, allow you to satisfy the distance matrix you come up with. So this procedure involved in create a Gram matrix. Gram matrix is almost the same as this, okay? They somehow interchangeable. So if you have a Euclidean distance matrix, you can change that into Gram matrix. If you have a Gram matrix, you can transform and get Euclidean distance matrix. But why we need to introduce Gram matrix? Because Gram matrix is much easier to compute. It's just dot vectors of these points. So you just, if you have a matrix contain those data points, you can just do a dot product between all the pairs of dots in your matrix and get a Gram matrix. Well, distance matrix is a little bit harder to compute. You have several other steps, but they're the same information, contain the same information. That's why we use quite a lot, use this inner product, dot products between data points to create a Gram matrix. Gram matrix is like a very useful matrix form, contains the information between distance or similarity between data points, and it can be easily computed and can be used for the downstream, for example, to do the multi-scaling, multi-dimensional scaling. So that's the way you convert Gram matrix to Euclidean distance matrix, okay, by doing this operation. Looks complicated, but in MATLAB you see this equation. If you have a Gram matrix and you do diagonal Gram matrix, diagonal transpose, that's you get a distance matrix. That's what I'm talking about. Okay. So at the end, we will use that information to reconstruct the map. Okay? Oh, by the way, if you want to use Euclidean distance matrix and to get the Gram matrix, that's a source code, that's operation you can use. Depends on your next operation. If your next operation is based on Gram matrix, then you want to create that. Or you already have this already, and you want to use that, you convert to Gram matrix. If you don't, maybe you want to use a Gram matrix because it's very easy to compute. Much faster, okay? So this is a so-called classical multi-dimensional scaling. And this function takes Euclidean distance matrix into as an input because you know the distance from one CT to another. But internally, you need to do this formula to convert that to Gram matrix and take eigenvalue decomposition and return this, and then you reconstruct your map and give you the position of those cities in on this map. And this piece of code is to do this. Give you a distance matrix. You found the best solution for arrangement of those cities on the map. Next, very important matrix is called covariance matrix. We just mentioned here how to compute, and we will do that. I think for the dimensionality reduction, we still have to revisit this, how to compute this. So here, the big X is a matrix, contains multiple points, data points. Then you transpose this matrix and then multiplicate the matrix by itself. Then you get the covariance matrix. You need to be clear and what is your sample, which is your feature variable. By the way, this is unrelated to this matrix because that big X is your data, not the distance between data, okay? Nothing to do with this. Remember, you have X matrix, that's your data, okay? You need to know, in data science, we're talking about the samples and the features. Features means those variables or values to describe each sample, to give you the characteristics of those samples. So if you think of this as a samples, and then you have feature variables. So we think each row is a data point, okay? So because this is somehow transposed, not by column. It was by always we say by column, each column is a data point, and then rows by center. But you have to know you have to know which is variable to describe which sample. So if you study single cell, and we focus on the cells, so those are the cells, those are the genes. Okay, have to be very clear which, because the covariance matrix at the end, covariance matrix is a feature by feature. It's C, okay, or sigma. So if you have 6,000 genes to describe your cell, your covariance matrix will be gene by gene. Unless you want to study gene as your sample, so you can transpose this. Say, I want to study gene and use the cell expression for this particular gene to describe the feature of genes. Then in that way, on your one side, you consider genes as your sample. And then the cells and their expression is your feature variables for the gene. That's your gene-based study. Here we use a cell-based, cell-centered, centric study. We need to compute covariance matrix. It's gene by gene, okay? So how can we get gene by gene results if we do multiplication of this X? There are two ways we can do X times X transpose, or you transpose X times X. So which way is correct formula to do matrix multiplication in order to get feature by feature matrix. So selection is, this is 1, formula 1, this is formula 2. So if your data input is like this, which formula you should use to get that? Yeah. So this is the correct one. The second one should be used. You want to transpose your data first, become a matrix of a gene by cell, then multiplicate the cell by gene. Then at the end, you get a gene by gene matrix. So really it depends on your input. If your input is cell by gene, you use that. If your cell is gene by cell and you want to get the gene by gene, then you do the opposite. You do, it's already transposed. Anyway. So what's inside here? Basically, they're dot products between genes, okay? So each of this Cij is a value. So for the gene, so this is the coordinates between gene I, gene J. It's pairwise for all the genes, pairs. So this could be the gene I, this could be the gene J. Okay, their relationship is built into this covariance matrix. It's very easy to compute, right? So you just transpose your matrix and time by itself. You get a covariance matrix. It tells you the relationship between those genes, whether they are co-expressed or anti-expressed. That information is built in there. And normally, statisticians will divide by n-1. N is how many genes here. So gene and so number of the features. Somehow take an average, because if you have more genes, then your covariance values inside here become larger. But you normalize it so you can compare between maybe cross data samples. Okay, so this is a special distance that's defined by Mahalanobis. So we call him Mahalanobis. He's an Indian mathematician. This is very similar to Euclidean distance. The difference is in this formula. This term are the same as Euclidean distance. This is to compute every data point to the center, the distance. This mu bar is in the center. And adding a covariance matrix in the middle. And this is the inverse covariance matrix, and to compute the distance. The feature of this distance is if you compute, for example, a distance, this is center, okay? So that's a mu here, the point. That's center of this cloud. And if you compute the distance from here to here, from this point to here,
or from here to here. So the value is not very different from a Euclidean distance. That's what you see on the plot. However, if you compute the distance from here to here, so this d, the value will be much larger, will be increased. So like 1, 2, 3, those data points are the outliers. They are stand out from the overall cloud of this trend of this data points described, which information has been encoded inside the covariance matrix. And this 1, 2, 3 will have a highest Macaulay-Nobis distance from them to the center of the data point than any other data points. Although the Euclidean distance maybe looks like this one from here is really long, but because it's follow the trend of your data, Macaulay-Nobis distance will weighted lower by the information inside the covariance matrix. So the distance from here to the center will be shorter actually from a 2 to here. Okay, questions? Yeah, each point is your sample, yes. Yeah. Yeah, so this particular distance is just use the information, the background information about your distribution of your data, rather than just directly compute without considering any others. If you use a classical Euclidean distance, you just compute, okay, the bar is like this, and it's shorter than this point to here, but in reality, we want to pick out this 1, 2, 3 as outlier from other part of the cloud. So this will be very useful somehow. By the way, if you cloud of points, just randomly, there's no pattern and no direction, it's just a random cloud, then the Euclidean distance and the Macaulay-Nobis distance will be very similar to each other. So the distance of this will be similar. However, if your data point is distributed along this axis, and then this pink distance is much longer than the purple. So in order to compute the Macaulay-Nobis distance, you need to do this. Okay. Once you see this next time, you know it's doing the covariance matrix. It's this formula here. You get the relationship between your feature variables. We will use this quite a lot. Okay. Yes. Cell Ranger's report. As you said, you already know Cell Ranger generated this HTML report and gave you very basic matrix to judge the quality of those basic matrix. You want to check number of cells, coverage, saturation, those values. In this sample, estimated number is a little bit low, and here is a better sample, but the average rates per cell, basically how many rates have been mapped into each cell. We want this number to be higher. This would be so let's see what's the number of reads. We want to reach 20,000 normally, but so in this case, this sample a little bit lower than that. This is over sequenced basically for some reason and only get so few cells, but the sequence is allowed per cell. we want to know the median genes per cell. So cells, then you count UMI. If a gene, the UMI equals 0, then you don't think that the gene has been expressed in that cell. So in order to have that gene to be detected, you at least have to have this cell has to have a UMI greater than 0, okay, 1 or above. So you want to count how many genes you can detect in each cell. This is a median number, so some cells may have 10,000 genes you found non-zero values. Some cells maybe only have, say, 1,000 genes. In that case, the number is just too low. We want to have a downstream control to get rid of those genes based on how deep they sequenced. If you have a cell, say, I sequenced 25,000 rays in there, but only detect 900 genes. For some reason, this cell is just not normal. And yeah, we later we will have some control to get rid of those outlier cells. Okay. Another number I want to look at normally will be percentage of the rates you map the genome. So this is 80%, this is 90%. Okay, so if you map human gene sample rates into mouse genome, this number may be lower than 75% because you use the wrong reference genome. And you probably will consider what happened, the quality of your reference, whether they are subspecies, they are huge genome sequence change. Another number you want to look at the saturation. Yeah, so to me, this sample looks under-sequenced. So you need to increase some kind of a sequence depths. But still, you already have so many cells. I really don't know. So you really have, based on the experience, use system there somehow. Something is hard to explain, but if you have many samples, look at other people's report, maybe you figure out. As I said, you look at the curve. You should have a sudden drop somewhere. If you have a flat drop, that means your cell sample, the sequence library environment contains a lot of environmental mRNA in there. So even some empty droplet contain this environmental mRNAs still have the information considered as a half cell, and then you have a very slow, gradual down curve, then this is not good. We need a clean cut. This needing, yeah. And yeah. UMI. Yeah. Yeah, in this case, there you have a lot of cells, but that's right, yeah. Unique UMI, UMI distributed in genes. That's right, yeah. Yeah, so it's all the signal against the background, okay, to change the threshold, consider. So this indicates the quality of the barcode sequence. So you want this number to be higher. That's indicated the sequencing quality. So if your Illuminer didn't success, then at the very beginning of the rates, maybe the quality too low, it gave you the errors when you do the assignment. If the quality is low, because your barcode is only how many 12 base pair, so if you have a say 10% of errors, then the 12 base pair may have a one A you read as a C, so you change your UMI or barcode, then you make the wrong assignment of the cells. I don't think you can do that again. Yeah, so the error rate, you want to have a more in this Q30 maybe means 0.001 errors. Again, you have to know how to look at this curve. This is a log, and then this 1, 10, 100, 1000, 10K. Again, this 10, 100. So this is a log form plot. We want to see this kneeling. Okay, we want to find this kneeling. Yeah.
So when we go back to the slide where they're comparing, so when the reads per cell is higher and the number of cells is lower, that is 2022.
Mm-hmm.
If we look at the sequencing saturation, it's 72.5%, whereas sequencing saturation is 10.3%
Mm-hmm.
15,000.
Mm-hmm.
So the mean reads per cell is lower. Is it because of that the sequencing saturation is low?
It's possible, yes.
Because all other things have increased,
Mm-hmm.
while only the sequencing saturation has gone down.
It's possible linked, yes. Maybe you sequence more, this will go up a little bit. This coverage certainly will go up, yes.
What is sequencing saturation?
I assume it might be number of cells that the sequence completely or map confidently on the genome. I assume, I don't know. It's the first time even I'm sequencing.
Yeah, I don't know the definition, so we have to look at the technical documentation of 10x genomics product in order to understand exactly how they calculate this, how they come up with such saturation. Yeah.
Because we recently sent out for sequencing and we received the data like 10 days ago. So even we were trying to understand because the other people who we are collaborating with, they suggested that we do some more dissections even though we sent like 120 genes.
Mm-hmm. You want to do another round of sequencing
Yeah,
to increase.
that every round increases like the consolidated number of reads.
Mm-hmm.
So we got like 35,000 cells approximately.
Mm-hmm. 35,000 cells per sample. That's no, several.
Estimated number of cells was I think 35,700.
Oh, that's a lot. Then. Yeah, that's too much.
Yeah, so
Yeah.
we were trying to make sense of it.
Yeah. So this is like a raise. I record say 20,000 will be better. Yeah. But this is acceptable maybe, and you need 25,000 genes detected per cell should be normal.
So apparently,
Yeah.
sequencing saturation is meant to tell you the library complexity and how much it's captured. So you have the UMIs, and then it looks at that without amplification bias, and then it takes into consideration the fraction of confidently mapped, the valid cell barcode UMIs that are non-unique, and then it looks at that and gives you a percentage based off of how much of the library complexity that they were likely to find.
So this image is to show you, it's not a single cell, but it just recorded the neuron firing in the brain of mouse. And I want you to appreciate this similarity between the gene expression. If you consider these are the cells and the density of this light indicate this cell expression. So I want to have you have an impression that the cells is not always constantly express one particular genes, but it's always like sparkling. So among the population, you will see this firing events across different cells. So gene expression itself is a stochastic event, and we should consider transcription bursting. So cells at the same sometimes express that particular gene really high, and sometimes it's shut down, just take a break. So if you have the genes working like that, if you look at a population of cells, then you will see a lot of those. As a group, you see, okay, maybe 50% of cells suddenly express this gene really high, another 50% seems to do nothing, and that's normal. So you detect your cell in your sample and see, okay, a particular gene 0, and then suddenly 200 and 0 again. That's in your population. So don't be considering the stochastic as your enemy. Actually, you should use this as your friend, because that's a feature, the nature of the transcription. And those noise is more important than the average. I will show you later. So I against differential expression analysis in single cell. I've been in different symposiums talk about this. Biologists always use average change. See, I have to upgrade my gene expression for this gene